Okay. Pick up where we left off, shall we? So we should have a pretty good chance of finishing 12 and getting a healthy start on 14. That's the morning section did. You guys are slightly ahead of them. So we already talked about polygenic inheritance, right? So we talked a little bit about how this is a violation of the one gene, one trait component of the Mendelian pattern, right? And so human height was an example, right? So basically, if you have people line up according to height, you can see that basically what's going to happen is they're going to have some very few short people, very few tall people, but a lot of people that are right there in the middle, right? Basically creating a natural bell curve. And that's the reason why they call polygenic inheritance continuous in nature, right? Because that bell curve is, the name of it is continuous. It's a continuous distribution, right? That's what it's called. And it's actually makes a lot of sense, right? Because basically continuous describes the nature of the data, right? So instead of having just categorical data where you basically leap from 4 to 5, right? From 5 to 6, you've got basically every potential data point in between 4 and 5, right? And so that's kind of what continuous data is. So height is an example. I think I left you guys with skin color, which was an example, right? That has 8 genes to it. I think that was about the last thing we did before we scrambled out the door desperately. So the next one is also a violation of Mendel. In this case, this is a violation of the one gene, one trait thing as well, but in a slightly different way. Pleiotropy is a situation where you have one gene that has multiple traits or that's involved in multiple traits. Now, generally speaking, whenever you see pleiotropy, usually what you're looking at is a disease gene, right? So the examples I have here is cystic fibrosis and sickle cell anemia. Cystic fibrosis is a good one because this is basically a disorder that's a pretty catastrophic disorder. But what this is basically is a mutation in a chloride channel. That's it. It's just one mutation in one protein that happens to encode a protein that forms a chloride channel, just like we saw with facilitated diffusion. But as it turns out, you actually have multiple symptoms. Generally speaking, every single symptom set is considered a different phenotype. So like if you take a look at cystic fibrosis, you'll see that one of the things that you would suffer from is a bunch of liver disorders. That's basically this one right here. So these are all variation, different types of liver issues that associate with that. Pancreas is also, you have some disorders there associated with digestion and some absorption as well. You have some respiratory dysfunction. So your cilia don't have the capacity to actually sweep your mucus up out of your respiratory tract. So essentially, you can't actually cough stuff up. So when a particle gets caught in your mucus in your respiratory tract, it kind of sweeps up and you kind of cough it all up. You can't do that because your cilia don't have the capacity to sweep that up. So what happens to it? It just kind of stays there. And the cilia, by the way, is what gets your phlegm, right, your mucus, up into your cough zone, right, which is a type of reflex, right? So you kind of get up into that cough zone, and then that's where you kind of have that uncontrollable urge to cough. That's because it's moved up into that zone. The idea is you cough it all out, and then you're good. But if it can't sweep it up there, then it never gets up into that cough zone. And what you get is this basically dislodgement, this accumulation of mucus in your respiratory tract, and you can't get it out. Right? And that's a bad thing. Sweat glands, this makes a lot of sense, right? Because it's a chloride channel. One of the things your sweat does is it basically accumulates and you sweat off and excretes salts. Of course, in this particular case, you would imagine that you would have a toxic buildup of chloride because you can't move it around the body normally. And so you would have an excess of chloride in your sweat as well. Each one of these is referred to as a specific phenotype. Or a trait. phenotyper. So one gene, multiple phenotypes, right? So basically every single system, organ system that you affect is considered a new symptom or a new phenotype in this particular case, or a new trait. So that's pleiotropy. The next one we want to take a look at is we want to attack number 2, right? So we've kind of been picking on criteria number 1, right? One gene, one trait. Now we want to go after the 2 allele system. And of course, the first one is multi-allelism. And this is basically a situation where a gene has more than 2 alleles in the population. Notice I didn't say in the individual, in the population. You don't ever have more than 2, but the population at large can have more than 2. That'd be a multi-allelic system. The best example of multi-allelism that we're going to take a look at is the actual ABO blood grouping system. So where you have 3 different alleles there. We'll take a look at that because ABO system is also a really good example of another type of pattern that we see later on. And what we're going to do is we're going to put this off until we get both that one in, and then we're going to take a look at both of them together in the ABO system. Okay. So the next piece is going to be picking on criteria number 3. That's the complete dominance, right? So remember we said you have to have complete dominance, but what if you don't? Well, there's 2 different scenarios where we don't have complete dominance. The first one is where you have incomplete dominance. And this is where the dominant allele doesn't completely mask the recessive. So there's some bleed through. The other violation of this is what's called codominance. Sounds similar, but it's not the same. This is basically where there are 2 dominant alleles that express equally. And we're going to take a look at that actually in the ABO blood grouping system, right? So those are violations of number 3. Now, when we take a look at incomplete dominance, we're going to take a look at incomplete dominance first, and we'll take a look at an example of incomplete dominance. And then we'll take a look at the ABO blood grouping and then sort of cluster together multi-allelism and codominance and take a look at those together in the ABO system. So in incomplete dominance, we've seen this one before, haven't we? Because we started the chapter off looking at what? Red flowers and white flowers. And of course, when we cross them, what do we get? Pink. So that's a type of incomplete dominance, right? So dominant red is not strong enough to completely suppress the white. So there's a little bit of white bleed through as evidenced by the washing out of the red, right? So that's a type of incomplete dominance. When you take a look at incomplete dominance, this is kind of like our example. These are 4 o'clock plants. A couple things I want to draw your attention to. Notice it looks very similar, doesn't it? Doesn't it look familiar? Basically, you have the same parental F1 and F2, same basic setup, slightly different results. So let's take a look at this first of all. First of all, let's start off with the notational differences. In genetics, there's a couple of different notational differences that you'll see. The most common that we see, especially in just kind of beginning genetics, is the uppercase, lowercase letter version of it. That's the most common one you see. But like if you were to look at a researcher, they do their notation slightly differently for very specific reasons, right? So for instance, plant people will do their notation a little bit different than fly people, will do a little bit different than yeast people and so forth. And that's just because of the nature of the types of information that they're trying to get across, right? In this particular case, this kind of shows us a particular type of notation system where you start off with kind of a stock term, in this case, C, which stands for your phenotype, which in this case is color. And in the superscript, you basically put the individual allele, right? So in this particular case, if you've got a red version of color, then that is going to be superscript R. If it's a white, then it's going to be superscript W. A lot of times, especially like in a research lab where you're generating a lot of mutations of a particular gene because you're studying them, a lot of times you'll have lots of different mutations. And so using upper and lowercase letters is just not specific enough, right? Because if you have a mutant, which would usually be a lowercase letter, that doesn't help you, right? Because not all mutants are the same. So one mutant could be mutated in this area of the gene. Another mutant could be mutated in this area of the gene. So the mutants could be fundamentally different breakdowns in different areas of the gene. And so as a geneticist, you want to study what those implications are within that gene. So to just say lowercase letter doesn't give you enough information. So a lot of times what will happen is geneticists will use this notation system. And so they'll use some sort of a stock number for whatever the phenotype is that they're looking at. And in the superscript, a lot of times, especially if you have like, maybe a dozen different mutations that you're looking at, you'll just use a number. You'll just use 1, 2, 3, 4, 5, and 6. And so you'll just keep track of your mutations by number. And we see this a lot in human genetics. We see this in especially a lot of labs where they're dealing with lots of mutations and are trying to study different breakdowns in this particular gene. So it's a fairly common approach to use this. So in this particular case, we're going to start off the same way we did in the semester. We're going to take basically a true breeder for red, right? Sounds very similar. Basically, this is what Mendel did. You're going to cross it to a true breeder for white. Well, in this case, instead of what Mendel got in F1, what did you get in F1? You got your heterozygote. That was the same, but your phenotype wasn't, right? In this case, what you got was pink instead, right? So this is incomplete dominance. Why? Because the red dominant is not sufficiently strong enough to essentially dispatch the white. The white is still bleeding through. Well, you still then follow along the same pattern as Mendel did, right? You take your F1s and cross them to each other. In this case, it would be pink to pink, right? And you generate the same F2s. So basically, you have your same red gamete with a white gamete for one parent, a red gamete with a white gamete for the other parent. When you put the two reds together, you're going to get a homozygous dominant, right? When you put the red and the white together, you're going to get heterozygous. And when you put the two recessives together, you're going to get white. Notice what I just circled there is my genotypic ratio, yes? One homozygous dominant, two heterozygous, one homozygous recessive. Nothing different there. Exact same thing that Mendel saw so far, right? But where's the kicker? The kicker is in the phenotypic ratio. What did Mendel see? He saw 3-to-1. What do we see in incomplete dominance? We see a red, half of them are pink, and a white. So what we see is 1 to 2 to 1. So in incomplete dominance, your phenotypic ratio, which is 1 to 2 to 1, 1 red, 2 pink, 1 white, and your genotypic ratio, 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive, are exactly the same. So note one thing, you did the exact same thing Mendel did, the exact same experiment, but you came up with different results. Why? Because it's a different pattern of inheritance. There's a slightly different set of rules here, right, that affects dominance. And so this is one of the reasons why Darwin, who was the one working on snapdragons, failed. Because number one, he failed to reduce the problem to its simplest level, which Mendel succeeded at. But number two, he was coming up, or he would have come up with ratios that he probably didn't know what to do with because he didn't know what the comparative was, right? So this also kind of highlights something as well, is notice your ratios are always used, like in genetics, we're always trying to create these crosses and we try to get these ratios. And the ratios are telling us what pattern of inheritance or what set of rules these genes are obeying. And it tells us what the patterns are, right? Because depending on the pattern, you'll have different outcomes, which is important because then when you get to somebody, some family who has some hideous genetic disorder segregating through their family, and they want to know what's the likelihood of me having a kid with this. You want to be able to figure out what the pattern is that's happening in the family. That means you have to understand what rules that gene is governed by. And then you'll be able to tell them, well, this is the pattern it's following, which means that your likelihood is going to be this. Okay. And then, it's all decision making after that. Hard decision making, by the way. Just like that. Okay, so now let's take a look at the ABO system, right? So this is both multi-allelic and codominant. So multi-allelic, let's take a look at the alleles. So first of all, this uses that same convention. So you have the root notation of I. I actually stands for immunoglobulin. And you have multiple forms, right? So in this case, you have IA, which is a dominant allele. You have IB, which is also a dominant allele. And you have little I, which is a recessive. So you have 3 alleles. And this is in the population, right? So any individual will only have 2 of these. And of course, that's dependent on what your parents have. Okay. So that's a multi-allelic system. And of course, we have different systems that have more alleles than that. There's no rule, actually, that says you can't have an infinite number of alleles. There's nothing that says, oh, you can only have 3, right? You can have as many as you want. We actually have some gene systems that have quite a few alleles in them. Of course, obviously, you would imagine that would really kind of mix up the bunch, right? So the more pieces you have to pick out of the genetic hat, so to speak, the more chances are that you're not going to get the same choice twice, right? So that's kind of mixes up the population quite a bit. The other one is codominance. And so basically what codominance essentially means is that both A and B are equally dominant to each other. So they're equally expressed and seen in the phenotype itself. Now, they're dominant and codominant to each other, but fully dominant to the I, little I. Right, so the I is a traditional recessive. So let's take a look and see what we're talking about here. So just by the way, this is the genetics piece of it that I told you about that you want to pair or that you will pair along with your Mendelian stuff that we did in the lab, right? Because remember we did blood typing, but we didn't do that until lab 15. And there was kind of like a little bit of a genetic sort of a discussion in there about blood typing and stuff like that. Well, that's going to be filled in with this one. So what you want to do is, and it's not like super like, oh my gosh, I got to be like a professional level geneticist to figure this out. But what you want to do is you want to take the stuff that you learned about in the genetics lab, right? The monohybrid, dihybrid stuff and all that sort of stuff that you guys work with, and then pair it with your blood results with the genetics in it. So you want to fuse those together so that you kind of have a full body view of not just the monohybrid, dihybrid genetics, but also the blood typing, the reading, but also the genetic consequences of that, right? Because that's how we basically determine like identity by blood type. Like, is it possible for you to be my father? Well, no. Why? Because the genetics, right? It's not possible for me to be O type when you're AB, right? Because I had to have gotten a little I from mom and from dad. So if dad is AB, that means he has no little I in there, right? So those are the sorts of things. And it's just kind of like just a little bit of on the light touch of genetics. So let's take a look and see what we're talking about. So remember in lab, when we covered this, we basically said that your blood type is essentially glycoproteins that are on the surface of your red blood cells, and they have slightly different forms. In the A type, you're actually making a carbohydrate that is galactosamine. That's the actual carbohydrate you're making. And so that has a particular form to it. And then, of course, we all know that if you're essentially your immune system is growing up with this particular form, then it learns how to tolerate this, right? But it rejects all else. What is the all else? Well, the other form is galactose, right? So that's the other carbohydrate that's out there. So that's the B-type form. And so an A person would make antibodies against galactose, right? Because it doesn't know that. It didn't grow up with the B form, so it's foreign to it, so it'll kill. The B-type person will make antibodies against galactosamine, right? Because that's the foreign thing to it, because it didn't grow up with galactosamine, it grew up with galactose. So the galactosamine looks crazy to it, right? So it'll attack. In an AB person, you have both galactose and galactosamine. So what antibodies you're making here? Yeah, you know what everything looks like, right? You grew up with A on one side and B on the other, right? So you guys were all neighbors. So you're feeling good about this one. Notice also that this one is codominant, right? So notice I made certain to add as many galactosamines as galactose. Notice it's equal expression. There's not one over the other, right? And so that basically is what dictates them as codominant. And then, of course, with O-type blood, basically what you have is no carbohydrates on there, no galactose, no galactosamine. So in this case, what kind of antibodies are you going to make? Yeah, everything looks crazy to you. So you're just going to you're going to attack it all, right? So you're going to attack it all because that's what you don't know. Huh? There probably is, but at this point in the semester, and in the week, I'm too tired to try to figure it out. But so if you come up with it, lay it out there. But so first of all, one of the things I want to sort of dictate is these two columns right here are your phenotypes. So this is what's called, so your phenotype, we usually think of your phenotype as sort of what you see in the mirror, right? That's the simple version of it. But your phenotype can actually be broken down on the molecular level as well. Okay, so in this case, in your terms of your blood type, I'd refer to this as your physiological phenotype. Physiologically, this is what you are. You're an A-type person or a B-type person or whatever. The actual carbohydrates on there would be your molecular phenotype. You're actually looking at what the molecules are as a consequence of, by the way, your genes, right? So the reason why you're making galactosamine or galactose is because of the genes, your genetics, right? So that's the reason why this is a molecular phenotype. So a lot of times students will miss that because we get so caught up in like hair color and eye color and all this sort of stuff we can see. We just think of like, oh, your phenotype is what you can see. Well, it's actually what you can't see as well, right? Your phenotype, technically speaking, is any physical manifestation that is associated with the expression of your gene, including your molecules, right? So this would be considered a molecular phenotype. Your blood type would be considered a physiological phenotype. So then in that case, what's the genotype? What's this column over here? So if you're an A-type person, then your genotype can be one of two possibilities, right? So you can either be homozygous dominant or you can be heterozygous. Right? So you can either big A, that'd make you an A-type person, or you can be a big A, little I. Notice that is complete dominance relative to little I. Okay. Same thing with B. You can either be big B, or you can be big B, little I, heterozygous, and that will also make you B-type. Notice again, B is completely dominant to little I. Now, an AB person has one A allele and one B allele. But when you take a look at the phenotype, you notice that there is equal expression between the two. That makes them codominant. So neither one diminishes the other. They are equal instead, eye to eye, exact peers. Right? So that's what makes them codominant. And then, of course, little I is your recessive, and that's the O-type person. So when you're looking then at transfusions and the idea of immune attack and rejection, if you're an A-type person, can you take B-type blood? Can you take AB blood? No, because the B part would trip your immune system, right? Can you take O blood? Sure, because there's nothing scary there, right? So an A-type person can take its own, right? Or O. Now, can they give blood to a B-type person? No, because it'd be rejected, right? Can they give it to an AB person? Sure, right? And of course, they can give it to an A person, right? So basically, you can give it to either an A or an AB because they both have A in common, right? Or you can take it from A and O because there's nothing scary about O. Okay. Let's take a look at B. Can a B person take it from A? No, because their immune system doesn't know what A is, right? Can it take it from AB? No, because the A will trip their wires. Can it take it from O? And of course, they can also take it from B, right? So you can take this from either B or O. But can they give it to A? No. Can they give it to AB? They can give it to AB, right? Because AB knows what AB is. It had part of his B, right? Can they give it to O? So they can only give it to AB and themselves, right? Now let's take a look at AB. This one, you have both of them on there. Can AB take A blood? Sure, right? Can it take B blood? Sure. Can it take O blood? Sure. Can it take AB blood? Well, of course, because that's what it is, right? So basically, AB can take it all. It's what we refer to as the universal receiver. So if you're an AB person and you're hemorrhaging, just walk in there and say, hey, let's line them all up. Just load me up. Take everything off the shelf. I can take it all. Don't even need to test for it. Just load me up, right? So it's good being an AB if you need to get a lot of blood. However, can they donate very well? Can they give to A, B, O? I mean, okay, nobody can give to O, right? Because that's just because they have nothing. So everything looks scary to them. So they can't really donate very well, AB can't, but they can receive quite nicely. Now let's take a look at O, right? Can O receive either A, B, or AB? No, because they all have a problem, right? They can only take from O, but can they give to them? Sure. So this is their universal donor. So essentially, we all love you O types because you can essentially donate your blood and you can save us all. So if you're O type, don't worry if you see a black van cruising behind you suspiciously with blackened out windows. And if you wake up in a dark alley somewhere with kind of the missing time phenomenon and a faint taste of cookies in your mouth, it was an alien abduction. They don't feed you cookies. It was just bond fees. So they just needed a couple of pints. So you O-type people, they basically track you through your fillings. So they don't do microchips like dogs. They just put those chips into your fillings because, well, most people need fillings or vaccinations, right? They just inject it that way. They think they're giving you MMR. No, I'm just kidding. I freak students out every now and then when I do that one. It's a lot of fun, actually. You're like, oh my gosh, are you serious? I can see, I do that for a reason. I try to see how many of you guys are actually. Yeah, it's like, did I got you? I think I got you. I got a couple of you. Yeah, I can definitely. Some of you are following me down the pathway here, so I'm just gonna keep going here. Kelsey. So that obviously that's a little extreme, right? So, however, I did that once. I didn't do quite the fillings one, but that was an addition. I love that one, actually. That's one of my favorites. And vaccines is actually my favorite. That's going to replace it because there's so many anti-vaxxers out there, just freaks people out. So it's just like really messes with them. But anyway, I just say that's not nice, is it? But it's fun. It's fun. It's entertaining. And I do this so many times a week. I've got to figure out some way to enjoy it. And this is how I do it. I mess with you guys. Just hope you go with it, right? You guys are like, great. So we're just guinea pigs here. You're just kind of messing with like a cat with a mouse kind of a thing, right? So anyway, no, but actually I did that once with the black van one anyway. And actually one of my O-type students were like, dude, I know you were kidding, but you're kind of not that far off. Yeah, because it's like, bond fees, they're like all over me, like all the time, because it's like they keep track of the O-type people and they're aggressive about keeping tabs on you because they know that you can donate to anybody. And it's like, he's like, I mean, you name it. You want to be on the Christmas list, they'll put you there. Hanukkah, they'll put you there. Kwanzaa, you're there. I mean, you name your holiday, they'll put you on it. You want to create one, they'll put you on it. They'll do whatever you want. They'll feed you as many cookies as you wish. But yeah, he said like, yeah, they are a little over the top. Yeah, that's what they want. They just, it's like, hey, it's like, here's some cookies. Just give us a couple of gallons. It's like, you don't need all that blood. I mean, yeah, it's like, seriously, come on now. I mean, you can make it. We can't make it. You can make it. Drain you until you pass out, and then we'll just let you recover, right? So, and we justify it by giving you cookies. which if you think about it, is kind of mean, right? Because it's like, wait a minute, I just feel like I just got robbed, but you know, man, the cookie's really good. So now I'm confused, right? Good, bad, good, bad, or good. It depends how you look at it, right? So I mean, it could be good and good. It all depends on how you view needles, right? So yeah, for me, it is clearly good and bad. So I'd be like, you know what, keep your cookie. I'm going to go to the store and get my own. You're not getting near me. Luckily, I'm not O-type, so nobody cares about me. But anyway, you're like, what type are you? Like, are you trying to say pass moral judgment on blood types? Like, no. Okay. So this is an interesting one because I don't know where to put it, but it is actually interesting. So this is basically looking at environmental impacts. So let's take a look at our kitty. So this is basically a situation. Now, up to this point, all of our patterns of inheritance have largely been purely genetic, right? So these are genetic rules that govern segregation of genes and the patterns with which they segregate. Now, for the first time, we don't just have a genetic thing, we also have a non-genetic thing in the environment. And so you can see then how this actually affects your phenotype. So you have basically this idea of your genetic plus your environmental equals your phenotype. Now, this is kind of another layer of complication of phenotype, right? Before it was just like you are what your genes say you are. Yes and no, right? There's more to it as well. In this particular case, you have to have a genetic component, but that genetic component is interacting very strongly with an environmental reality. And an environmental reality that changes. So you get variation in your phenotype. Now, by the way, whenever we take a look at this, and this is kind of what makes genetics fascinating, because we realize as we sort of look at our relatively simple view of our phenotype, when we look in the mirror, when we get into it, we realize, whoa, wait a minute, there's a lot going on here that we don't really think about normally, right? There's more than just genetics. There's like a lot of other variables going on here as well. Whenever we get to scenarios where your genetics is actually equally complicated, like for instance, what happens if the genetic component is actually polygenic? And what if you have lots of genes involved in whatever this particular phenotype is, right? What you're talking about is multiple variables. And if you overlay on top of that, lots of different environmental realities that affect those genes, then you get your phenotype. You know, this is the reason why, remember when I mentioned the idea that our reality is basically filtered through our genetic filter? What I mean when I say genetic filter is exactly this. Your genetic component, all of this genetic complexity and all these genetic variables are basically washed through these environmental cues that are constantly changing, so that what you get on the outside of that is essentially your genetics that has been variably washed in the environmental non-genetic cues to create a very complex and ever variable phenotype. And we get into a lot of that in the genetics class where we kind of start getting into those variables. Yes, I will never miss an opportunity to plug genetics. So just, right, I got a plaid suit on and I've got the broken down used car over here that I'm trying to sell. So only it's not a broken down used car. It's actually like a Bentley, right? But people don't think it's a Bentley. It's like a Porsche, but they think it's a Carmen Ghia, right? It's like, no, they look the same, but it's not the same. This is actually a Porsche, not a Carmen Ghia, right? For those of you car, you know what I'm talking about, right? So the 70s Carmen Ghias are like, wait a minute, does that look like the 911? Yeah. But you paid a lot more for the 911, didn't you? It's much better. Yeah. So that was for you car fans. But anyway, so if you have a lot of variables like this, then we start to move into another world where we start to refer to things as multifactorial. So that's basically where we see like there's just so many variables going on here. It's a really tangled, twisted mess, and it's difficult to separate out the variables. A good example of this is like human behavior. A lot of human behaviors will exhibit familial patterns. So we can see that there is a genetic pattern in there. Now, some of these familial patterns are very weak. So we can see it, but it's just a hint of familial descent in there, right? But the problem is, of course, with human behavior, there's an awful lot of environment and learning that goes along with that as well. And so human behaviors and psychological phenomena, things of that nature, are oftentimes very multifactorial because there's a lot going on there. And what you have is essentially this big gamish. It's like this big goulash of genetic variables and non-genetic variables all mixed together in this bubbling brew, and that's you. Isn't that lovely? Kind of very appetizing, right? If you like goulash, then yeah, it was cool. But if you don't like it so much, then it was a little disgusting. But either way, that's us, right? So here's a good example, a very discrete one, by the way, to kind of start off simple. And we're looking at pigmentation in Siamese. Now, I'm not a cat person, but if I ever got a cat just to look at, just because I wouldn't want it near me, I'm just not a cat person, it's just more of a dog person. I prefer dumb and cute as opposed to master of the universe, and enslaver of all, because that's what cats are. They're out to basically enslave the human race, and they kind of do a little bit. Yeah,
they do a good job.
They do, and their owners love them for it, right? So dogs, well, they're dogs. I mean, you don't have to go any further than that, right? They're just like, wow, you really do need us, don't you? Because you would be screwed on your own, right? A cat is like, yeah, I got this covered. I'm just humoring you, right? So that's
awesome.
Yes, exactly, right? We're the lap dog for them. That's basically how that works out. So, but anyway, Siamese are awesome. I mean, of all the cats that are out there, I think probably Siamese are my favorite, just because they're just visually stunning. I mean, if you look at it, those piercing blue eyes. I mean, that's that right there is like, wow, okay, that's cool. Because that's rare in animals. That's like super rare, right? So when you see that in an animal, you're like, wow, that's like really cool. Okay, let's take a look at pigmentation. So pigmentation, melanin production, is actually driven by tyrosine, the amino acid tyrosine. So what happens is tyrosine is basically converted into several precursors. It eventually gets sort of modified into melanin, right? And one of the enzymes at the top of this melanin-producing pathway is tyrosinase. Now, tyrosinase, basically, when you have temperatures that are above 33 degrees, so hot, just to give you a reference, body temperature is about 37 degrees. So this is a warm area. If you have it hot, then it's going to inactivate tyrosinase. Tyrosinase will then not create the melanin precursors, and you will get no pigmentation or no pigment production. And so you see that in the warm areas of the body, minus pig. Okay. And so these are the hot areas of the body, right? So these are basically too hot to actually activate tyrosinase. However, in cooler temperatures, below 33 degrees, it will activate tyrosinase, which will then actually make pigment and deposit it in those areas. So where are those areas? It's where we associate the cold areas of our body, right? Which are our extremities, our feet, our hands, right? Our ears, the extremities of our body. And you can see the same thing is true here. So you see you got cold areas in the feet, the ears, and the face. And so because of the temperature effect, then you will be depositing pigment in only those cold areas. Now think about this. So you're thinking, okay, that's great. I can see where the temperature of the environmental piece comes into play, but what about the genetics? Well, tyrosinase is an enzyme that is encoded by the tyrosinase gene. So the gene itself makes tyrosinase. That's the genetics piece of it. But the tyrosinase is not activated until it meets the environmental piece. So it's like the genetics is one half of the story. You make it with genetics, you activate it with environment. So what that does then is you have two different locales of control. You can control pigmentation by either controlling how much tyrosinase you make, that's the genetic control, or you can control it by the distribution of heat, that's the environmental control. So this basically then creates a much more complicated and much more variable phenotype for the cat. So the one thing I always like to sort of mention is why, right? I mean, other than like, okay, you're a stunning cat, you're going home with me, and you're going to basically eat until you drop, right? Of course, that's a good thing. But like, why in the why, right? Well, a good example of like I like to use instead of cats is Himalayan. Oh gosh, is it Himalayan? The rabbits. It's like the white, not the snowshoes. They're mostly white, but they've got black ears and they've got a little black face. I think they're the Himalayans. I'm not sure. I have to double check on that one. But here's a good example, right? Because you're in the snow mostly. That's where you live. That's your thing. That's your habitat. So the question is, why in the world would you possibly want black ears in a snowbank and a little black face in a snowbank? Think about it. Is that a good idea? No. First of all, snowshoe hares, Himalayan rabbits, things like that. Yeah, the Himalayans. Thank you. Right. So they got the little black muzzle and the little black ears. Raphael's going to have to walk around the room and show it to you guys. Right. Right. So if you think about it, if you're a rabbit or a hare, you are at the top of everybody's menu. You are item number one on the McDonald's list, right? Every predator in the world has you right at the top. And you can actually see this in prey species, like prey pets. If you have bunnies, like they spend their entire life, even if you're cuddling on them, they actually don't think you're loving them. They think you're trying to eat them, right? That's just the way prey species work. It's very different than predator species, which is what the major pets are, right? Dogs, cats, those are all predator species. And so they kind of have that predatory sense about them. But if you have a prey animal, it's very different. I mean, I just walked to the room and my rabbits would be like, I mean, they'd swear, this is it. He's going to eat us now, right? It's like, it's really bad because it's like, they're so cute. You want to cuddle them, but then you realize it's like, wait a minute, this is only really working for me, isn't it? It's like, you're terrified. I can tell you're terrified, even though I'm just like hugging on you or something like that. It's like, you're just absolutely scared out of your mind because you don't see it as cuddle time. You see it as like, okay, this is it. I'm going down, right? This is, right? Right. It's over. Yeah. It's like literally. So that's the way they think because that's what the reality is. They're at the top of everybody's menu. So here's the question. You're in a snowbank and you've got these black ears and this black face. Why would that be a good idea? It'd be, yes. So it looked like a bigger face, a different face, right? It would be confusing to the predator. So that's definitely a strategy. If you confuse them, butterflies do the same thing, right? So basically you put big old eye spots on your wings to confuse predators because they're like, wait a minute, I see all these eyes all over the place. So where are you exactly, right? But the other thing is also the same thing that butterflies do is it's also avoidance. Think about it. If you're a predator, what are you most likely going to go after? Well, small things, but what do you see? If you have a Himalayan in the middle of a snowbank, what are you going to see? A little black blob with two black lines above it. So if you're not confused about what this is, what are you likely going to go for? You're going to go for what you can see, the black, right? So you're going to likely go after an ear as opposed to the kill shot, which is the core. So if you think of this cat as a prey, and I was a predator, then what am I likely going to go after? The tail, the leg, the ear, but the kill shot is right here. So survival-wise, the strategy of this phenotype is to basically confuse and to deflect. If I can get you to attack my ears, I'm going to wiggle them, then you're going to be off target, right? We have a lot of that actually around the city. How many of you guys like squirrels? They're cute. I mean, when they're not chewing on like your radiator belt and stuff like that, I mean, and chewing up your wires in your car, they're okay. But no, they're cute, right? Because the thing I love to watch squirrels is they're really animated, aren't they? I mean, you see them running around, and what's the thing that's moving the most on a squirrel? Yeah, the tail by far, right? It's big, it's bushy. The first thing you see is what? That big old bushy tail, right? And they got that thing flicking around, right? And then if you scare them, what do? They kind of freeze, but what's that tail doing? They got the S shape and they're like, right? They're wiggling that thing. What are they doing?
They're trying to make it seem like
Yes, it's avoidance, right? Because the idea is if you're coming after me, I'm going to freeze the kill shot, but I'm going to move my big, fluffy, visible tail around because you're likely going to go for my tail. That means I'm free. If you look closely, there are a lot of squirrels in Denver that have half a tail. In some cases, no tail. So basically, their tail is serving them well. That's exactly the same thing the Himalayan is doing. If I can get you to miss the kill shot, then I've got a chance. That's exactly what this pigmentation is doing. So you can see that when you're developing phenotypic variation, there's clearly a very strong sense of environmental adaptation. There's a reason behind it, right? It's not just like, oh, hey, let's just be stunningly pigmented today, right? It's like, no, there's a purpose behind it, and it's usually associated with one of the big biological questions of life, right? Stay alive long enough to reproduce. That's it. For what? Yeah, I mean, generally speaking, dark colors do absorb more heat. But like in terms of people, it's probably not as significant because even though the skin looks dark, the layer of melanin is actually not as, it's not like clothing, right? So it's not like in order to heat up, you would actually have to absorb the heat and sort of trap it in there. But the problem is your skin is actually better at shedding heat than absorbing it. So even if you had like super, super dark, like if all of your skin pigment alleles were all dark, right? You were like that outlier, the really, really dark side, like really, really dark. You still probably wouldn't be dark enough to get that sort of dark clothing absorbing of heat effect because your body is still infinitely better at shedding heat than it is absorbing it.
How about the pigment on fur?
For the fur, well, the fur is different, right? Because the fur pigment is generally not for heat absorption, although you do get a little bit of that. But most of that is actually air trapping. So that's actually how an animal heats it up, heats themselves up. So ideally what happens, it's kind of the same thing happens with us, right? The reason why we wear clothes and why they're warm is because our internal body, our body's shedding heat, but it basically gets trapped in the air layer between our clothes and our body. That warms up the air layer there, which makes us warm against our skin. Same thing's true for an animal's fur, is they're warming up the air that the fur is trapping against their skin, and that's what's keeping them warm. In addition to like fat layers and all sorts of stuff. So it's a little bit different. So I mean, pigmentation, absorption of sun, lack of absorption in a lighter organism is probably a minor component of it, even though technically the physics is still the same. It's just not as big a deal as the physiological heat management processes.
It's more of like, you know, when foxes turn colors based on the season, I guess.
Yeah, and then birds do the same thing. Generally speaking, that's not usually a thermoregulation issue. That's usually a mating thing. So it's usually, I mean, rabbits do the same thing. I think the snowshoe hare does the same thing. So we usually think of it as sort of like white all the time, but it's not, right? So there's some of those rabbits that will turn white. Arctic fox, I think, does a similar sort of thing, right? So they're white in the winter because they're camouflage against the snow. But then in the summer and spring months, it basically changes its fur color out and it kind of gets more of a mottled kind of a brownish color to match its environment. So that's kind of a slightly different variation. It's not usually a heat related thing. It's usually a camouflage related thing, honestly. Or for birds, it's a mating thing, right? So you kind of put on your fancy tux to impress all the ladies like in the spring, and that's kind of what you do. And then once you've done that, then you lose all that and you kind of turn into just like a brown bird in the winter. So it's like winter birding is tough, right? Because you don't have all that mating plumage to rest on. It's like, wait a minute, aren't you supposed to be like yellow? It's like, yeah, but that's a new step, right? Because this is winter. Okay. Now let's take a look at a gene interaction. So this one basically gets back to a violation of D. That is the randomness of the segregation. And that's epistasis. This is where one gene affects the expression of another. Notice in this particular case, we didn't talk necessarily about the segregation, right? So that would be the genetic thing, because up to this point, we've been talking about how do genes segregate. Now we're talking about expression. That is to say, we're not talking about how they segregate, but now we're just talking about how you see them, right, in the phenotype itself. So now we're really talking about how does this affect the phenotype itself. And so that's epistasis. So Emerson worked with corn and did an epistasis. And so it essentially did the same thing that Mendel did, right? The dihybrid cross, working with two different genes. And then what he saw was a 9:7 ratio purple to white in his corn. But notice what 9:7 is. 9:7 is a variation of 9,331, right? So if you take 9 and you add the others together, you get 9,7. So that's what Emerson saw. We're going to take a look at what Emerson's data looks like here in just a second, because it is kind of an interesting little, it's a little more complex than just basic epistasis. So I want to kind of introduce, since we just talked about pigment, I want to talk a little bit about a pigment version of epistasis, and then we'll take a look at Emerson's, which is kind of like a little bit of a notch beyond that. So let's imagine we have a pigment gene, B. And big B means you're going to be making black pigment. Little b means you're going to be making brown pigment. So there's your B gene. Okay. Now we have another gene, E. Big E basically means that you're going to make pigment. Little e means don't make pigment. So if I have that right there, what color pigment am I going to make? It's going to be black, right? However, what happens if I put the E gene in front of it like this? It's a heterozygote. It's still going to be black, right? Okay, now let's go ahead and do this one here. What color is that going to be? That's going to be brown. Now, what's that one going to be? What would it have been if it was just that, right? But because of the little e, the decision that's made first is do not make pigment. Don't care what color you're going to make because it's irrelevant. I just made an initial decision that we're not going to make pigment at all. So it's irrelevant what genetics you have for making the color. So in this case, it's going to be white, the absence of pigment. And so that's basically how epistasis works. So it's essentially a decision flow, right? So it's like there's one decision that had to be made first in order to make the next. So in this case, the E gene had to permit the B gene to express. The B gene will only express upon permission of the E gene. That is to say, E is epistatic to B. So here's an interaction, right, between these two, where E affects the expression of B. And of course, that'll change your phenotype, right? Black, brown, or white. Now let's take a look at Emerson's corn. So basically, Emerson did a cross, did two true breeders, got purple in the F1, then crossed these guys together, got basically your dihybrid scenario. But here's what Emerson saw. In cases where he saw a dominant in A and a dominant in B, he saw purple. So your dominant A, dominant B, you saw purple. Dominant A, dominant B, saw purple. Dominant A, dominant B, saw purple. Dominant A, dominant B, saw purple. But now you have a dominant in A, but now you got a recessive in B. Now what do you see? White. So what that basically means is that B is required to be dominant in order for A to express. Doesn't matter what's at A, does it? If B is recessive, that trumps anything that's at A. So what this means is that B is epistatic to A. So you can see you got a dominant in both, so you're purple. B again is epistatic to A, so it's white again. You got a dominant in both, so it's purple. Now you've got A is recessive, but B is not. but you see white, which means that A is required and seems to be epistatic to B. Again, A is epistatic to B, so it's white. You have both dominants, it's purple. You have one dominant, B is epistatic to A is epistatic to B here, and well, here they're kind of like both attacking each other, right? There's nothing going right there, but these are all white. So what does this seem to suggest then? This seems to suggest that B and A require each other to be expressed. That is to say, they're epistatic on each other. Now, what we found out later was that actually what we saw here were two components of a pathway. So basically, you started off with a precursor molecule that was colorless, that was converted into a colorless intermediate by enzyme number A, which was encoded by gene A. And then enzyme B, which was encoded by gene B, would turn this colorless intermediate into a purple pigment. Now, in cases where there was no B recessive, then it didn't matter how much A you had. You could make all the colorless intermediate you want, but you're never going to get to the second step because B has to be functional in order for this to finish. In cases where you didn't have an A, it didn't matter what happened with B because you would never actually accumulate intermediate. So B would never have anything to work on because A, you never got out of the gate. Right? So in this case, both A and B needed each other in order to get expression, which is purple. Okay. So you think of this as epistatic to each other. B, what's happening at B affects the expression of A. What's happening at A affects the expression of B. So they're basically working against each other. So that's like double epistasis. So for you table people, this is just your table, right? So you basically have polygenic inheritance, pleiotropy, multiallelism, your dominance, right? So you have incomplete and codominance in there, your environmental factors, and your gene interactions. By the way, there's lots of gene interactions. There's epistasis is just one of them. And so in genetics, there's like, we go into a lot more depth into all the different types of gene interactions you can get into. So there's a lot of different things that are going on in terms of gene interactions and what they do with each other. Okay. So that's the end of 12. So I know what you guys are thinking. Maybe that's it. He'll stop talking about genetics. Oh, you should know better than that. I've just begun. All right, I'll be talking about genetics pretty much until December. But wait, am I going to be here? Yes, you are, because you'll be in genetics. He says ominously, right, as I follow you with a black van. Okay. Yeah. It's like, you will join or die. It's like you guys be like, yeah, I like to say he's passionate about genetics. A little scary, but yes, there's definitely passion there. Yeah, dangerously passionate. But anyway, so now we've got chapter 14, right? So this is basically going to be looking at DNA, right? So remember in chapter 10, we basically were talking about the cell cycle. We talked about G1, SG2, so forth. And we said, well, S phase is duplication, or duplicate and separate. And we said that basically duplicating your DNA in the S phase, we're going to hold that discussion off. Well, now the discussion is here, right? So this is a story of what's going on in the S phase. So the mechanics of how you go about duplicating your DNA. But of course, as everything, we all start with a little bit of a primer on our basic history, right? You guys are like, got more history in biology than I did in history, actually. So first of all, the important thing to understand is what we had with Mendel and kind of where we left off, right? So remember, Mendel was looking at information, not at stuff, right? We think of chromosomes, we think of mitosis, we think of meiosis, we think of all these things because it's in our textbook. But remember, Mendel had no concept of genes, of chromosomes, of mitosis, of meiosis. He didn't know any of that. He didn't even know about gametogenesis, right? He probably wouldn't even be familiar with the term gamete itself, right? So what he was looking at purely was information flow. So really, a lot of the hardcore stuff, like the structural stuff, didn't come until the 20th century. And it took probably a good half a century before we started making some headway on this as well. So when we get to Mendel, the end of Mendel, what are the questions, or what's the dominant question that's left over? Well, the basic question is, what is a gene? Or in this case, as Mendel saw it, what is the particle of inheritance? Now, this took a little bit of time to figure out. And here's the reason why, because remember, at this particular point, like at the turn of the century, like in the 1900s or so, the aughts, if you will, right, from 1900 to 1909, if that's what you like, right, that's gun talk, 30-06, that kind of thing.
Oh.
Right, so, but this is an area, remember, we're just discovering stuff. So we don't really know a whole lot. At this point, we're just largely, like we're doing the 4 o'clock plant crosses. That's what we're doing. We're doing corn crosses. That's kind of what we're doing. But we don't know anything about molecules. We're still pretty fuzzy on the kinds of molecules that are out there. We have a notion of chromosomes. We've seen them in microscopy, but we still have a lot of questions about that. We don't know nearly as much as you guys know about chromosomes at this particular point. So even though we knew of some things, we had a lot more questions than answers, right? So it's always important to remember that filter, because when we come into 1928, a famous experiment done by Frederick Griffith, we actually start getting some traction on this. Now, what's been happening for the first almost 30 years or so of the century is largely just kind of playing around with the Mendel structures, trying to figure out, like, you know, what does my system look like? And remember, we had major avenues. Some people were working on fruit flies. Some people were working on different types of plants, 4 o'clock plants, corn, things of that nature. Some people were working on humans. So there was different types of things going on, and they were all running into their own specific limitations, because each model organism has a separate set of realities that you have to deal with, and they're different from each other. So this is kind of what we were basically struggling with for the first 30 or so years. Frederick Griffith, however, came along, and he was going to be one of these more human-centric types. And he was basically studying Streptococcus pneumoniae. So Streptococcus is a genus name. We usually associate that with strep throat, right? This is not the strep throat one. This is the one that causes pneumonia. So there's multiple species under the Streptococcus genus. Pneumoniae is one of them. Strep is a different species. So it's a different thing. But this is the bacteria that basically will cause bacterial pneumonia. Which, if you're thinking about this particular era, now, just to kind of give you a little bit of a calibration on kind of where the world was at this particular point, just to kind of give you a bit of a societal sort of a perspective on the realities of who we were at this particular point. World War I was over, right? That ended in about 1919. And Germany was busy making reparations for that. Which was not very pleasant. So 1928 actually represents probably a very tumultuous period of time in Germany. At this particular point, Hitler was a public figure. In about 4 years or so, Hitler would be elected chancellor. But the Weimar Republic is falling apart at this particular point under the heavy weight of these reparations from World War I. At this particular point in the US, we were basically coming to the end of the Roaring 20s. So after World War I, it was good times. I mean, it was, if you want to know what it was like, read The Great Gatsby. That is a novel set in the Roaring 20s. That's 20s era stuff. So lots of prosperity, lots of people getting rich. It was good times all the way around. But this was also an election year. This was the year when Calvin Coolidge stepped down as our president and Herbert Hoover won. Of course, we're only a year off from one of the biggest moments in U.S. history, which was what? The Depression, right? Or in this case, actually, the stock market crash, which happened in 1929, right? And then that's basically is the reason why Hoover became a terrible president, because he was the unfortunate sucker who happened to be riding the ship when it went down, right? So not that he did anything wrong, he just, just very unlucky, right? However, that's kind of what was going on here. So you can imagine sort of the nature of it. Depending on where you are in the world, you have a very different experience at this particular point. One of the things we also noticed that this is pre-antibiotics, right? So if you got pneumonia at this particular time, it was basically just, bundle up, stay warm, kiss the family, and start praying, right? Because there is nothing we can do for you. And that was basically it. And pneumonia is a nasty customer to dance with. Even by today's standards, pneumonia is no walk in the park. You can imagine that pneumonia back then was basically a death sentence in a lot of ways. So there was a lot of reason to get your head wrapped around this, right? And that's kind of what Frederick Griffith was doing. Frederick Griffith was basically trying to understand this bacterium so that we could basically learn some information so we can beat it. Right? That's kind of what fuels biomedical research. Is biomedical research doesn't, what we see is the development of a therapeutic. It's like, oh, you're developing a drug. Well, yeah, that's great, but that's the end of it, right? Before you get there, all biomedical research rests on basic science. In order to beat it, you have to understand how it works first. And that's where Frederick Griffith was. And the idea is I want to understand how this works so that we could potentially figure something out to beat it. So basically what he's noticed is that with Streptococcus, you had two different strains. You had a virulent strain, basically which causes disease. And then you had a non-virulent strain, the R strain, which doesn't cause disease. So it's very interesting, right? So you see this, two different strains. And so obviously the question is, how does this work exactly, right? I want to understand why these two strains exist, what their role is, and how they interact with each other. So basically then what Griffith did was he worked with mice, he affected mice under different treatments, and he basically tried to determine what was going on with these two different strains. So here's what he did. The first thing he did was he took the S strain, infected his mice with it, and of course it's a virulent strain, right? So basically what he got was a bunch of dead mice, right? Okay, we expect that, yes? Then he infected his mice with the R strain, or that's the non-virulent one. And so in this particular case, what did he see? Living mice. Okay, so far so good, right? Everything checks out. But now what he wanted to do is he wanted to treat it. So here's one of the ideas that was rattling around them, right? Because you're talking about a hospital. You can imagine what hospitals were like in this time. They're probably filthy pits, actually. They're kind of filthy pits now. If you think about it, you don't ever hear of a staph infection outside of hospital contact. That's also true, right? Because, well, that makes sense because the hospitals are where we go when we're sick. We spread all of our germs there. Nothing good happens there. And so we just get things that we normally wouldn't get access to, right? So they've always been that way. They're that way now. You can imagine how much worse they were back then. So you can imagine then there's a lot of motivation to try to figure out, okay, wait a minute, how do we manage to sort of put a damper on all these germs that are out there, right? In this particular case, pneumonia, right? Well, one of the ideas back in those days was to heat it, right? Just like you do with mountain water, right? You boil it to try to heat kill all the bad guys, Giardia. So the idea was same here, right? Well, what if we heat killed the virulent strain? If we could kill it, then we'd be like sterilizing the tools. It wouldn't matter, right? Well, there's two different things that Griffith basically identified. First of all, he said like, well, wait a minute, there's killing it and then there's removing it, right? So there's two different scenarios you can imagine. We could either kill it and just leave it there, or we could kill it and remove it. Those are two different things, right? So if your idea was to heat kill it and to go use it on somebody else to cut on them, guess what? You're still spreading the S strain on that person you just cut. It's just been heat killed. But what if that person has R strain on them already? What happens when you mix the two? That's what he wanted to know. So what he did was basically, he first of all started off by heat killing the S strain. When he did that, he had happy little mice. Makes sense, right? Kill the virulence, and then you get happy mice. Now, here's the thing. Now he realized that, well, wait a minute. In reality, these two strains are going to be mixed. If we heat kill it, Eventually, if we keep using it, it's going to get mixed with other strains that are alive. Obviously, we know that the S strain's a problem, but imagine we just had R strain. What happens if the two mix? Well, here's what he did. He heat killed the S strain and mixed it with live. What did he get? Dead Mickeys. Wow, that is a head scratcher, isn't it? Think about it. You've got a dead body that somehow infects a live body and makes it dead, right? So it's kind of like, this is a head scratcher, right? Because then all of a sudden it's like, wait a minute, wait. You mean we can't just heat kill it and we're off and we're good? You're saying we have to heat kill it and remove it because we can't have it mixing with the R strain. Otherwise, we're just going to rekindle the virulence. So that's kind of what it looked like in images. So you can see basically you've got your S strain, which you inject, kill the mice. R strain, which you inject, mice live. Then you heat kill your S strain, inject it, mice live. Good, so far so good, right? Looking promising. You're about ready to go to the FDA for clinical trials. You're getting ready to put this one out on market, but then all of a sudden, what do? Mix your heat killed S strain with your R strain, and then all of a sudden, whoops. I'm glad I noticed that before I released it on the market, because then I'd get all sorts of lawsuits because I basically wiped out half the population. Okay. So what's going on here? Well, what Griffith basically found out was that there was actually seems to be some sort of a mechanism to transfer the information for virulence from the dead S cells to the living R cells. Now notice, at this particular point, he didn't know what that was. All he knew was that whatever was governing the information that causes virulence is somehow being transferred from the S strain to the R strain. He called that transformation. You're able to transform the R strain into virulence because of this information swap. Now, there's two different types of transformation that we see in biology. One is in cancer biology, where they talk about the transformation of a tumor as a tumor progresses from benign to malignant and to metastatic, right? There's a transformation to the next ugliest form. That's not the same word or mechanism or process as this one. This transformation is the ability for one cell to take up DNA from another cell, which is something, actually, as it turns out, is something that is very unique to bacteria, and that bacteria have the capacity to do, which turns out to actually be quite a handy dandy little trick that bacteria do. It helped us in research a lot. Question?
The S strain is the death.
Yes.
It doesn't mean that they become alive.
They're still dead, which means there's some sort of a mechanism to basically take whatever that information is and transfer it somehow into the R strain. What that is, at this particular point, we didn't know.
When the mice is retested, they find live.
The R cells are virulent now. So they look like they've been transformed into S cells, living S cells. Right. And since typically in these days, scientists weren't given to the theological concept of resurrection, we assume that those dead S cells that we killed are still dead. Somehow the R strains were transformed into S cells. So then if you fast forward to 1944, right, notice that we're a couple of decades further on now, right? So you can see how the progress grinds along, a couple of decades per major landmark discovery. So the pace of knowledge is much faster now, largely because these guys were kind of on that kind of slow growth curve, and now we're on the exponential side of things. But if you take a look at this, right, so in 44, just to give you an idea, 44, basically the war was not over yet, right? 45, it ends in 45. So we were still fighting. We're getting close to the end. At this particular point, Europe was a mess. London had largely been bombed out of its skull and survived, which was amazing in and of itself. At this point, I don't remember when the bombing of Dresden was, but at this point, we are starting to tighten the noose between Marshal Zhukov and the Soviets moving in from the east and us moving in from the west, although they're moving much faster through the east because we had, unpleasant little battles like Bastogne and things like that to sort of survive. So it was a little bit more of a slower go, but that's what's happening in 44. We're getting very close. Clearly the momentum is on our side. Okay. And so, if you're wondering, because I never miss this moment. This, by the way, is also an election year. Who's being elected here? Maybe Roosevelt. Yeah. Yeah. FDR was pretty much in there for just about forever. Just about. Literally, this is actually his last term. Right. So it goes from 44 to 48, and then Truman got elected in 48 to 52. But this is momentous because this is FDR, like literally, I kid you not, this is not hyperbole. He was dying, and we still elected the man. This goes to show you what a titanic personality FDR had in the 30s and 40s. Because a lot of times we think of like Obama and how charismatic and like just crazy, like magnetic he was. He had nothing compared to FDR. I mean, people were still voting for FDR like decades after he was dead. I mean, my grandma was one of them. I mean, she never voted for anybody other than FDR, like even into the 80s. I mean, it was like crazy. I mean, the man had just this almost messianic like aspect about him over the country. And literally he was dying. I mean, he's dying of congestive heart failure. He was literally dying. He actually died probably within a couple of months, I think, after being elected. Democrat. He was Democrat. But in 44, we elected a dying man. It's crazy. So yeah, America has a lot of wackiness. We are a strange and peculiar people, at best, right? On our best day. But that's what's happening here. We're basically electing a dead man, a dying man, and we all love it. And then we've got kind of Truman sitting behind him. So it's like, so I don't know if people realize they were voting for Truman or if they just were that delusional that they're like, no, he's still alive. We'll just, like the Russians, we'll embalm him like we did with Lenin and just like stand him up there. It'd be like, that'll be our president for the rest of our life. But anyway, it's hilarious. If you ever get a chance to read that entire biography of FDR, it's a fascinating moment in history. There's nothing like it in U.S. history, like even remotely like it. It's actually an amazing sort of a thing. And as it turned out, we're lucky that he was who he thought he was because if he had been like a tyrant or a villain, he could have absolutely annihilated this country. So the fact that he was the great person and the great politician that he was, and he had, good intentions and he wanted to do good things for the country, we were lucky because we were absolutely so beyond stupefied by him that you literally could have just cherry picked us and annihilated us if you wanted to. So it was lucky. He's a fascinating character. The nephew of Teddy, basically, the Roosevelts, strong crowd there. So this is when Avery McLeod McCarty comes into play. And so what they wanted to do is they wanted to get to what the Hershey Chase is next, actually. So they wanted to get at what Griffith was getting at, right? So what they said is like, okay, fine. So this virulent thing, whatever it is, you don't know what it is, but whatever this is, it's got to be inside that cell. That much we know. So what we're going to do is we're going to take cell extracts, which is basically a fancy word for saying cell guts. So we're going to take these cells, we're going to basically turn them inside out, eviscerate them, take all the cell guts and put them in a test tube. That way we know that whatever that thing is that causes virulence, it's in there. So here's what we're going to do. At this particular point, we knew enough now about nucleic acids and proteins and carbohydrates and lipids to know that it was the information molecule, the genetic molecule was one of these four. At the time, there was a raging debate as to whether or not that molecule was protein or it was DNA. So what Avery McLeod McCarty tried to do was try to essentially sort of settle that argument a little bit. So what they did is they took their cell extracts, which obviously has mixtures of all these different things in there, and you don't know which one is the one causing virulence. So what they did was they basically removed all of the protein out of there. The idea being that if you remove all the protein, and if that is the inheritance molecule, or it's causing virulence, then you would imagine that you would also remove the transforming ability. What did they see? They did not destroy the ability to transform. So what's the conclusion from this experiment? It's not protein. But wait, there's a pushback on this one. That's nice and all, but are you sure? I mean, did you get all of it? Did you get all the protein? I mean, can you be sure you got all the protein? Can you prove to me with another experiment that you got all the protein? Think about it. We already learned about chromatin, didn't we? They didn't know about chromatin back then. We know that DNA doesn't exist by itself, does it? It exists wrapped around what? Proteins, right? So that question becomes even stronger now. It's like, wait a minute. Are you sure you got all the proteins? Because I'm not convinced, right? Prove to me that you got all the proteins, because even if you left one behind, that might have been it, right? So then they say, okay, fine. We're going to go ahead and do the reciprocal. We're going to then take a look at the DNA side of things. So what we're going to do is we're going to take these DNA digesting enzymes, right, nucleases, and we're going to send them in there, and we're going to basically chop up all the DNA. What's the idea here? If DNA is the informational molecule, then if you chop it all up into little bits, then what's going to happen? You're going to lose the ability to transform virulence, right? So they basically digested up the DNA, and what they do? They destroy the transforming ability. So what's your conclusion there? That DNA drives virulence. Oh, but it's the same thing though, right? Okay. See, that's a perfectly good question. So basically probably what they had is some sort of a biochemical approach to it. And biochemistry in 1944 was just very much beginning. So they probably had techniques where they thought they were removing all the protein, but they probably weren't getting anywhere close to removing all the protein. But then when you digest all the DNA, right, what's the question? Did you get it all? Yeah, are you sure? What about other nucleic acids? I mean, at this particular point, you kind of have this idea of nucleic acids. You kind of might know that there's such a thing as DNA and RNA, that they're different. But remember, this is pre-DNA structure. So we kind of know a little bit about DNA, but there's a lot of mystery associated with these nucleic acids. DNA especially, and we know the most about DNA, so we know even less about things like RNA. I mean, RNA, if we knew about RNA, all we would really know is that it's a nucleic acid. I mean, we wouldn't have any clue about its structure, its role, what it does, how many different types of RNA are in there. So we have very little knowledge about nucleic acids. So you're basically trying to tell me that all these mysterious molecules you're going to digest up. How do I know that your enzyme, your nucleases, actually digest all the DNA? What if they don't digest all the DNA? What if they only digest specific types of DNA, right? Do you have an experiment to disprove my complaints? Well, at this particular point, they didn't. However, what they did have seemed to suggest DNA, right? But you can see it's not as satisfying, is it? Because this actually marks one of the first forays into this discussion, that potentially DNA is a genetic molecule. But guess what? It's not nearly as slam dunk enough in order to convince most people, right? Because there's still like when you're looking at these cell extracts, we know what's in those cell extracts now, right? We know that it's either nucleic acid, that it's either proteins, it's carbohydrates, or it's lipids. We know what they are. In a lot of ways, we know nucleic acid. We know that it's DNA. We know that there's 3 different types of RNA, right? rRNA, mRNA, tRNA. We have an idea of what they do. We also know a lot about other RNAs, even short chunk RNAs that are regulatory in nature. So we know lots of little bits and pieces that are in those cell extracts, not to even mention the proteins themselves. So at this particular point, we probably thought of most proteins as largely being mostly enzymes. Probably didn't think of them as structural too much at this particular point, but we know a lot of that now. We didn't know any of that back then. So the cell extract actually was almost was this sort of magic potion And we knew that there was stuff in there, but we had no clue what was in there at all. So the cell extract idea actually had many, many more question marks than answers. So you can imagine that if you're an opponent to this idea, you could say like, what you're finding is intriguing, but there are way too many questions in that cell extract, right? We have got to understand that cell extract to a lot more degree to understand what we're talking about, because you're talking about a lot of variables there, and we don't even know what most of those variables are. So you can't really say anything with definition if that's the case.
We don't know for sure.
At this particular point, we don't know for sure. We do now. We do now, but not at this point. Okay. Because it took a long time to get there. So when did that definitive one come along? Well, that was Hershey Chase in 1952. Right. So 1952, you are looking at an election year again. I don't know why they did these in election years. I don't know if they did this on purpose, but every single one of them has been an election year. This is, by the way, if you're wondering, this is when Eisenhower was elected in 52. Just saying. The reason why this is kind of an important year is because from 45, the end of the war, on to about this time, this marks the baby boomers. This is the baby boom. You come home from war being shot at a good chunk of your existence for quite a few years. And then you come home and you kind of do the stress release thing and you have lots of babies. And then we call that accumulation of babies the baby boomers, who are all now getting old, in their 70s, which is the reason why the majority of the high density apartment complexes going up are senior apartments, because there is a hell of a lot more baby boomers than any of the rest of us on the planet. And they know it, unfortunately. So that's kind of what you are, right? This is basically the birth of the hippies, right? That entire movement that's kind of historic in the 60s, right? The historic 60s and all that. And the ones that are largely in control of the universe now, and have been for quite some time, by the way, the same. Now that I like that is definitely a hard slider. When I was looking fastball, no question about that. It's our lab exam is not this Friday, it's the next Friday. This Friday is our lab review. So it's kind of like an open forum lab, but good question. I love the execution. I always appreciate having a breaking ball right there in the middle of some predictable pitches. So when did that definitive experiment come along? Well, that's Hershey Chase in 1952. And so what Hershey Chase basically did was they basically looked at bacteriophages. And now what a bacteriophage basically is a virus for bacteria. So this is how it looks. So you have a bacterium, and a bacteriophage itself is basically going to be mostly only DNA and protein. That's it. There's nothing else in there, which is really helpful, right? Because these happen to be the two molecules we're arguing about. So the way a bacteriophage looks, and actually this is kind of important because so when you take a look at this, what you have is the beginning of virology. So as it turns out, viruses, even animal viruses, HIV, coronavirus, adenovirus, whatever you want, they all have a very similar pattern. They're all basically protein and nucleic acid, and they all infect in a similar sort of a fashion. So when we were studying bacteriophages, I mean, Hershey and Chase didn't know it then, but what they were actually doing was studying the mechanism of virology for pretty much every organism across the board. Here's how it works. Typically speaking, is a virus will have kind of a capsule-like structure made of protein, like a little collar piece, also made of protein, and some like little legs, also made of protein. Looks like a little lunar lander. And then in the middle of this is going to be your DNA genome. Okay. Now, when this infects the bacteria, what it does is basically it will dock onto the bacteria itself, like this. And then it'll inject the DNA into the bacterium. Now, once it does that, it will basically hijack the bacterial processes and essentially take it away from making the bacterial proteins to start making the phage proteins. So instead, you'll be making virus particles. So you'll be making little capsules, collars, and legs. And then, of course, you'll be making DNA as well. Then eventually, you're going to assemble all this, and then you'll lyse. That's why it's called the lytic cycle. And what do you release? Lots of little viral particles to go and infect other cells. And that's how basically viruses spread. Okay. Now, the important thing is the phenotype that you're looking at, the information, is the lytic cycle. So that is to say that hijacking of the bacterial machinery. Okay. So what you want to do is you want to correlate this with your inheritance molecule. So that is to say that the information for virulence should follow the molecule that carries the information. They should be correlated together. You should find them in the same place. So the reason why this is powerful is because, unlike other systems, this has a natural mechanism and a very thorough mechanism to separate the two. So what you knew is the information went inside the cell. And that's where the infection happens. That's where the phenotype is happening. The function goes inside the cell. So the question is, who goes inside the cell with it? Right? The information molecule has to go in there with it. So what is it? Is it protein going in? Or is it DNA going in? So what I gave you was kind of like a little bit of the punchline. We already know that it's a DNA, but this is actually what told us that that's what it was. Okay. So let's take a look at their experiment. So what they did was they basically just set up a system so that they can label the DNA and the protein specifically, separately. And you can do this. So in DNA, because DNA has the phosphate groups associated with the nucleotides, you can actually use radioactive phosphorus and the isotope of phosphorus, P32. And this will basically be radioactive. So what will happen is wherever P32 nucleotides are used in the DNA, it will highlight the DNA, right? So this will basically highlights the DNA. You can do the same thing with protein. Only in this case, you use an isotope of sulfur, right? Which is basically found in what? Cysteine. Remember, we talked about that early on in the functional groups where he said that the thiol group is part of the cysteine amino acid. Well, this is where it comes in handy. So proteins, most proteins will have at least a few cysteines incorporated in them. So whenever you actually add cysteine with this S35 isotope in there, this will basically highlight the proteins because they will incorporate the radioactive cysteine. So now you can differentially label DNA and protein. Then what they did was they tracked these radioactive molecules and they tried to figure out where did the phenotype go? Is it correlated with the protein or is it correlated with the DNA, right? So here's what they did. In this particular case, they're going to use S35. So you can see you've got the highlighted protein components of it. They allowed the phage, which was now highlighted, to infect the bacteria. And then they blended that all up, which separates everything, kind of makes a big goulash. Now what it doesn't do is it doesn't chop up the cells themselves, right? The cells may remain intact. It just sort of breaks off all the phages and all that sort of stuff. And then what they did is they centrifuge it. And when they centrifuge it, basically all the heavy stuff will basically go down to the bottom into the pellet. And then the supernatant is all the light stuff, which will stay in the supernatant solution above. So because the cells are intact, they're naturally heavy. So that means that they're going to spin down into the pellet. However, the phages are small and light, and probably a good number of them got chopped up during the blending. So you got a lot of protein chunks out there and relatively light and small phages. So they're not going to spin down quite as heavily, right? It's like trying to compress down feathers, right? They're really light. They're not going to compress down, not like sand. Sand is heavy. It'll actually condense down quite nicely. So what they noticed here was that the label was in the supernatant, but not in the pellet. Now, why is that significant? Because remember, they said that the phenotype they're looking for, the infection, the information for virulence is inside the cell because the cell gets hijacked, right? They know that. The question is, what's inside? Well, what this is telling you here is that when you spin this down, the label that you see is not inside the cell. So that means the protein did not get injected into the cell. Therefore, the protein cannot be the informational molecule. Why? Because we know that the information molecule has to be inside the cell because that's where the infection is. So then when they did the same thing with DNA, right? Because you always want to, you don't just want to stop there and say like, oh, see, that means that DNA is it. No, that doesn't. All that means is the protein's not. That's not the same as saying DNA is, right? So negative information does not prove anything positive, right? It's like doing multiple choice, like process of elimination, right? If you know it's not A, it doesn't mean that it's C. It just means that it's not A, right? You can't conclude that it's C unless you actually have proof to conclude that it's C, right? It's the same basic frame of logic. Now then what they did is they followed that up with basically highlighting DNA with P32. Now you can see the proteins are not highlighted, but the DNA is. And then they infected it. They got the infection inside the cells. They blended it up and they centrifuge it down, just like they did before. Now what did they see? What they saw was the label was inside the pellet. What does that mean? That means that what was inside the pellet is the informational molecule because that's where the hijack is happening. What's inside the pellet? DNA is inside the pellet. This one is much more compelling because you have physical separation of the two in very uncertain terms, right? I mean, there's no uncertainty here, right? So there's complete separation, but you have differential highlighting, and you got it correlating with a known phenotype. So basically what you have is you've got a criminal identification that you've now located at the crime scene, right? So now all you need to do is watch them do it in the act. So this is basically the beginning of essentially locking it all down for us and saying, okay, fine. After this one, we feel pretty comfortable with DNA being the molecule because it's pretty clear. Good. Mm-hmm. So that's Hershey Chase. So basically, when you take a look at DNA structure, this is kind of recapitulation of what we already learned in the macromolecules chapter, right? So you can see that nucleic acids basically are composed of nucleotides, which have phosphate groups on them. They have a nitrogenous base, which is made up of adenine, thymine, cytosine, and guanine. And then, of course, at that 3' end, they've got that 3' OH group, right? That's important, right? This is your growing end. And so that's that important piece. That's associated with your ribose sugar, your deoxyribose sugar, right? So that's basically an important component of your DNA structure. So here you can see it. So here's your phosphate group. Here's your nitrogenous base. And then, of course, you can see your 3' OH with your lack of OH there. In that case, it's a DNA. If you have an OH there, it's RNA, right? That's basically, this should look familiar to you. This is literally almost like a recapitulation of what we saw in the macromolecules chapter. And then, of course, your nitrogenous bases are basically broken into two different groups, right? So you have your purines, which are the double dog dishes, like Purina double dog dish, right? So you can see your two little double dog dishes there, which are adenine and guanine. And then, of course, your pyrimidines, which are cytosine and thymine, and of course uracil if you're RNA. Right? So these two get swapped out in RNA. So in DNA, you see ACGT. In DNA, you see ACGT. In RNA, you see ACGU. Okay. So those are true for both of those. Now, one of the things that I always tell students, first of all, the way that you remember which ones are purines and which ones are not is something that I actually got at UC Davis in my genetics class, which I thought was stupid. But I remember it. That's the point, right? So it worked. But he said, just remember this: pure silver taxi. Of course, back in those days, it wasn't too far removed from the Back to the Future movies, right? So you had the DeLorean, which was awesome, right? The first stainless steel car, that was pretty cool. So that's what I thought of, like when I saw, pure silver taxi is basically a stainless steel kind of a DeLorean coming up to take you from the airport. That'd be cool. So I thought it was dumb, but what did he say? He said basically it means pure, for purine, equals silver, which is elemental symbol of silver is what? Ag, which is adenine and guanine. What's the taxi? TC, which is cytosine and thymine. So these would be the pyrimidines. To this day, that's how I remember it. Pure silver taxi. That the purines are A and G. And then from there, it just all folds out. Why do we want to know this? Because we want to build a DNA molecule. So the first thing we need to do is understand our parts. Okay. So this is basically our parts. Now this is kind of like a little bit of a recapitulation, right? So here you can see you have your two nucleotides sharing their phosphodiester bond. So here you can see those two ester bonds that are basically connected to that phosphate group in between. So you have a sugar, phosphate backbone, right? So that's a common backbone for all nucleic acids. And then of course you can see your 3' OH down here, that's your growing end. You can see your 5' phosphate up here, right? That's your beginning end. So ultimately, your growing end, your next nucleotide is going to be added to your 3' OH. Why? Because your OH is going to be the one that's leaving. That's part of the water that's going to leave during your dehydration synthesis. Okay. So one of the things that I want to lead up to, and this is kind of getting to a little bit more because we're going to be talking about Chargaff's rules here in just a second. But I don't want to start this story because it gets a little heated and I want to allow enough time for heat. But what we're doing is we're beginning the journey, the trek to the discovery of the structure of DNA. So that's going to be the beginning of DNA structure. The journey to the discovery of DNA structure. This is where I want to start. So I want to stop here.